Assalamu alaikum. We'll continue in renal physiology lectures. Today we'll talk about the renal concentrating mechanism. This is one of the simple lecture, inshallah. So by end of today's lecture, we'll be able to cover the describing the tubular reabsorption of water. We'll explain the mechanism responsible for the concentrating and diluting ability of the kidney and the principle of countercurrent mechanism. And the countercurrent mechanism in kidney that will include loop of Henle, countercurrent multiplier, and there is the vasorecta, which is working on the countercurrent exchanger. And we'll explain the hypertonicity of the medullary interstitial fluid, including the role of urea, the effect of change in blood flow in renal medulla, and the role of transport property of the loop of Henle and collecting duct. At the end, we'll explain the mechanism of action of antidiuretic hormone and the role of aquaporine. As a start, we'll talk about the water reabsorption. So normally in the adult man, we have mentioned this earlier, 180 liters of a fluid is going to be filtered per day. So it's a huge amount of fluid going to be filtered. However, the urine volume going to be between 0.5 liter to 1.5 liter. So that means 1% of filtered water is excreted as urine and 99.4 is reabsorbed. So there is a lot of reabsorption. So water reabsorption is passive. Since it is the renal tubule segment, it will flow in osmotic gradient. So water is going to move by the concentrating gradient. So generally, when water intake is low, the kidney conserve water by producing a small volume of urine. So that's going to be hyperosmotic and or concentrated urine. When your water intake is high, when you drink a lot of amount of water, then the volume going to be hyposmotic or diluted urine going to be produced. So the normal individual, again, the urine osmolarity vary between 50 to 1200 milliosmol per kilogram of water. This is a general principle. The ability to excrete urine will vary in osmolarity. That requires solute to be separated from water at some point in the nephron. And you remember there is one part in the nephron where the solute and water is separated, whereas it is reabsorption of water, but it's impermeable to any other solute. And there is a segment of that nephron still impermeable to water, but it's permeable to solute. And that in loop of Henle. We'll start talking about the water reabsorption in the first segment in the nephron, which is proximal tubule. Here, the reabsorption of solute in proximal convoluted tubule is in proportion amount of water. That means the solute and water are not separated in this part of the nephron. So the proportional between proximal convoluted tubule water and the absorption of the solute occur regardless the excretion or diluted urine we have. So always the proximal convoluted tubule have the same proportion reabsorption of water and the solute. So because of this, the fluid will leave the proximal convoluted tubule as an isotonic. So now we'll talk about the segment where it is separated, which is loop of Henle. Loop of Henle in particular, the thick ascending limb is the major site where the solute and water are separated. So the excretion of both diluted and secrete and concentrated urine will require normal function of loop of Henle. This is where the loop of Henle is. So excreting of urine in general, the concentrating depend on the body state of hydration, and it's regulated by antidiuretic hormone. So in absence of antidiuretic hormone, the volume of urine could be about 23 liters per day with a concentration of 50 milliosmol per liter. So in the presence of ADH, the volume of urine will be about 500 milli per day with a maximum concentration of 1200 milliosmol per liter. So the excretion of hypoosmotic or dilute urine. The fluid will enter the thin descending limb. So we have already mentioned that it's leave the proximal convoluted tubule as an isotonic. So now we're talking only about the loop of Henle. and the rest of the nephron. So in diluted urine, the fluid will enter the thin descending limb of the loop of Henle from proximal convoluted tubule as an isotonic. The thin descending limb, which is high permeable for water, but less for solute, as sodium chloride and urea. So as a result, the fluid in this thin descending limb will go, as it goes deeper, it become hyperosmotic, and the water is going to be reabsorbed as long as the water is going to be reabsorbed. So the tubular fluid, the sodium chloride, going to be greater than the surrounding interstitial fluid. So it's going to be high concentration of sodium chloride inside the tubule. So the thin ascending limb is impermeable to water, but permeable to sodium chloride. As a result, as tubule fluid move up the ascending limb, the sodium chloride now is passively reabsorbed. And the net effect of this concentration is tubular fluid going to be decrease. The thick ascending limb in the loop of Henle is impermeable to water, but actively reabsorbing the sodium chloride from the tubular fluid, which thereby going to dilute it. So as a fluid leaving the segment is becoming hypoosmotic with approximately 150 millismol per kilogram of water. As it's entering the distal tubule and the collecting duct, it's going to be actively reabsorb sodium chloride in absence of antidiuretic hormone. The segment impermeable to water, so the osmolality of the tubule fluid further going to reduce because sodium chloride is reabsorbed without water. So fluid leaving the segment is hypoosmotic with 5 to 100 milliosmol per kilogram of water. Medullary collecting duct is actively reabsorb sodium chloride. This is what the urine has osmolarity as low as approximately 50 milliosmol per kilogram of water, and it contains low concentration of sodium chloride and urea. Now we'll talk about the concentration of urine. So to create concentrated urine or hyperosmotic or hypotonic, and antidiuretic hormone, the ADH, is needed in the presence of the hypertonic medulla, what we call it VOG. And the first 5 steps of producing diluted urine are the same as here. The difference is here in the 6th step, which is in distal tubule. What's happening here in distal tubule? So producing concentrated urine in response of dehydration, as the ADH increases, and as ADH increases, it's increased permeability of luminal membrane to water by inserting a new channel, which is aquaporine 2 water channel. This ADH will concentrate the tubule fluid from 100 up to 1200 milliosmol per liter by the end of the collecting tubule. And you can see it here clearly in the nephron figure. We'll talk now about the osmotic gradient in renal medulla. So the factors that create and maintain the high osmotic gradient in renal medulla are 2 countercurrent mechanisms: the countercurrent multiplication system in the loop of Henle and the countercurrent exchange system, which is in the vasa recta, and then the urea cycling and low blood flow to the medulla. In order to explain the countercurrent mechanism that occur in medulla, we need to know some physiological characteristic of the loop of Henle. So loop of Henle consists of 2 parallel limbs with tubular fluid following in opposite direction. Filtrate will flow into the medulla in the descending limb and out of medulla in ascending limb. The ascending limb is impermeable to water and reabsorbs solute from fluid. That will cause the fluid within the ascending limb to become diluted. The solute removed from the ascending limb tubular fluid will accumulate where it's accumulated in the surrounding interstitial fluid and raise the osmolarity. So here the osmolarity is high. So we'll talk first about the countercurrent multiplication. Countercurrent multiplication is the process which produces the hyperosmotic renal medullary interstitium. You can see here the figure It illustrates how the countercurrent multiplier system increases the osmolarity of the interstitial fluid and creates the osmotic gradient from cortex all the way to the papilla. So in the first step, the lobe of Henle is filled with fluid from the proximal tubule that is isoosmotic with systemic plasma 300 milliosmol per liter. The sodium chloride is going to be reabsorbed through the sodium-potassium 2 chloride cotransport. If you remember the channel, the sodium chloride is going to be reabsorbed through it along the length of the thick ascending limb. Since this portion of the nephron is water impermeable, removal of solute diluted the tubular fluid and make it hypoosmotic. The interstitial fluid, on the other hand, going to become hyperosmotic due to the deposition of the sodium chloride, and this is called single effect. Since the solute transport is separated from water transport, the thin descending limb is highly permeable to water. So water leaves the descending limb until it is tubular fluid has the same osmolarity as adjacent interstitial fluid. So the thick, if you can see it here in the figure, the thick ascending limb establishes an osmotic gradient along its entire length of 200 milliosmol per liter. As we move through it, then you can see the fluid is shifted along the nephron. It's introducing a new osmotic fluid, 300 milliosmol, into the lobe of Henle from the proximal tubule and ejecting dilute fluid from the ascending limb. The single effect creates 200 milliosmol per liter. The gradient occurs once more in the following step, and the process is repeated until the osmolarity of the interstitial fluid at the tip of the lobe of reach 1200 milliosmol per liter. It is simple, not complicated as it looks. It's just a matter of repetitive absorption and reabsorption and showing how this maintained through this mechanism. So next, we'll talk about the other countercurrent, which is the countercurrent exchanger, occur in vasa recta. So some 5 to 15% of the total renal blood flow will supply the medulla and the papilla over the vasa recta. So these capillary are important not only in the supplying the medulla and papilla with oxygen and nutrient, but it's also important in removing the water that is reabsorbed in the lobe of Henle and the collecting duct. The vasa recta react as a countercurrent exchange, which is shown here in this figure. So in vasa recta plasma exchange within the interstitial fluid, what's happened? It consists of descending limb and ascending limb. As the blood flow into medullary depth, it loses water and gains solute, so it's become hypertonic. As the blood leaving medullary area, this is reversed, so it loses solute and gain water, and it's become hypotonic with more water. So this passive exchange of salt and water will between the two limbs of the vasa recta and interstitial fluid going to be called the countercurrent exchange. How does this function of the vasa recta as a countercurrent exchange? So it supplies tubular cell with nutrient and oxygen. It's remove reabsorb water and solution, and it's maintain the osmotic gradient established in the medulla. Now we'll talk about urea cycling and medullary osmotic gradient. So urea is not synthesized in the kidney. It is generated by the liver as a product of protein metabolism. So the interstitial fluid of the renal medulla is critically important in concentrating the urine. The concentration of sodium chloride and urea increases progressively with increasing depth into the medulla. So during antidiuresis, around 60% of the solute will be deposited in the medulla and papilla by countercurrent multiplication as sodium chloride. The remaining 40% is urea. So urea concentration increases in the late distal tubule, which become permeable to water, but not urea in the presence of antidiuretic hormone. When the fluid reaches the collecting duct in the inner medulla, urea move into the inner medullary and papillary tissue by facilitating diffusion. A substantial portion of this urea dissipate from the medullary tissue and re-enter the loop of Henle, where it diffuses into the tubular fluid of the thin descending and ascending limb. This will provide a renewed supply of urea. So the urea recycle through the rest of the loop and distal nephron will reach the higher concentration in the late distal tubule and collecting duct, where it is once again available to diffuse into the medullary tissue. Now we'll talk about the blood flow to the renal medulla. So blood flow to the renal medulla is via vasa recta capillary, which branch from efferent arteriole at juxtamedullary glomeruli and run in parallel to the loop of Henle and in the collecting duct. The second peritubular capillary bed, which branches from the efferent arteriole at cortical nephron, surrounded the tubular structure and the cortex. Talking about antidiuretic hormone, ADH. Well, thank for Dr. Asili. I found those cartoons entertaining. So ADH or vasopressin act on the kidney to regulate the volume and the osmolarity of the urine. So when plasma ADH level are high, a small volume of urine is excreted. So it is antidiuresis, and the urine is concentrated. When plasma ADH level are low, large volume of urine is excreted, diuresis, and the urine is diluted. We have mentioned this. So ADH control water excretion and maintain water balance without altering an excretion other than substance. So the mechanism of action of antidiuretic hormone and the role of aquaporine is explained here. So principal cell in the late distal tubule and collecting duct, their target cell for antidiuretic hormone. When ADH bind to specific receptor for ADH, which is V2 receptor, in the peritubular basolateral cell membrane of these cells, of the principal cell, adenyl cyclase, that located within the membrane, is going to be activated and generate cyclic AMP. Cyclic AMP can activate protein kinase A, which trigger a cascade of events that end with incorporation of water channel aquaporine 2. So incorporation of aquaporine 2 into the luminal and the apical cell membrane will make the cell permeable to water. So when ADH level are low, aquaporine 2, what's going to happen to it? It's going to be re-internalization. And with this, we reach the end of the session. It's a quite easy session, inshallah, and good luck.